Hello viewer, today I will talk about biological mutagen. I will talk about different types of biological mutagen, their characteristics, mode of action as well as biological effects. So, some viruses, transposon and bacteria, they can disrupt the gene function and causes cancer. They are called as biological mutagen. So, biological agents causing the induction of the high frequency mutation within the genome are called as biological mutagens. General characteristics includes the disruption or loss of gene function. Some of the jumping elements are there which interrupt the continuity of the gene and they cause the antibiotic resistance as well. There are three main types: transposon, virus as well as bacteria. The transposon, by relocation and multiplication within the chromosome, causes the biological mutagenesis, and the viruses, by disrupting gene activity, causes cancer. Bacteria, by oxidative DNA damage, causes biological mutation. Other types, the plasmid are efficient to transfer genes within the chromosome of different types of bacteria. In case of high level transfer, some parasites and wild type flies can take up gene from one species to another. Then symbiotic transfer is observed in between mitochondrial and nuclear genome. Some genes are stealed from the mitochondrial genome to the nuclear genome. In 1943, Barbara McClintock have found that the presence of some jumping gene on chromosome 9 of the corn or maize causing change in alluron kernel color, for which she has got the Nobel Prize in 1983. Let's talk about the characteristics of transposon. Transposon are natural and ubiquitous genetic tool for gene transfer. They are the jumping elements and they can be transferred within different parts of the chromosome either or from mitochondria to the nucleus even. They are actually associated with the adaptiveness and they give the evolutionary changes. They may be autonomous or non-autonomous. Autonomous means having some enzyme activity like transposase by which they can move, or non-autonomous means lacking that activity, need the help of transposase from other portion. They are environmentally regulated. They sometimes called as junk or selfish DNA without contributing anything to the host genome. They causes the genetic variability as well as susceptibility for the disintegration as well. They are giving the genetic instability. They can sometimes kill the organism. They have different kinds of length. They are found within the prokaryotes as well as eukaryotes, including bacteria and mammalian cell system. They can be transferred from anywhere and when they are getting accumulated, give the composite type of transposition. They sometimes move via retrotransposition, that is the RNA mediated way. They actually sometimes give the repetitive sequence like sign or line sequence in case of human genome. They propagated via non-homologous REK non-mediated recombination. Sometimes they have the mutated gene activity and their action is very much tightly regulated. Types of transposon in virus, in bacteria, in case of yeast as well as maize, we can find them within the human and other plants. Let's talk about them individually. In virus, the mu phage is the longest transposon, okay, and they have 100 different attachment site and 5 BP repeated sequence, and they cause deletion insertion as well. Bacterial transposon either may be insertion sequence or IS element, bacterial transposon or bacterial plasmid. The first one, IS element, insertion sequence, carrying the transposase gene as well as antibiotic resistance genes. They may be two types, composite or non-composite. In case of composite, we can find two IS element on either side, and within inside they can actually have transposase gene and antibiotic resistance genes. But in case of non-composite transposon, on either side no IS element, only inverted repeats are there, and internally they can carry repressor, resolvase as well as transposase gene. Next, the bacterial different types of transposon, they are actually named according to the TN, and the composite one is usually found in case of TN10 and TN3 is the non-composite. The insertional sequence element, IS element, these IS element is very common, and this IS element is actually causes gene deletion, insertion and forms the lollipop like structures, and this insertion sequence is 700 to 1500 base pair long, carrying 9 to 40 base pair repeat, but the composite and non-composite transposons carry 5000 base pair of total genome content. Next, bacterial plasmid, they may be the fertility factor containing F plasmid or the resistance factor containing R plasmid. This resistance factor causes the antibiotic resistance. Yeast. Yeast contains Ty plasmid, T transposon in yeast. So, on either side LTR long terminal repeats are there and lambda element or delta element is present. They are actually propagated via retrotransposition and they bind GAG, POL, ENV genes. Maize. Within maize, AC and DS elements are found. DS is dissociator and AC is activator. DS is non-autonomous, having no transposase activity and which is supplemented by the AC activator sequence. Apart from which SPM and DSPM, that is dissociator of the suppressor mutator, is found and they cause insertion, translocation, deletion. Apart from which DT locus is the mutated gene locus. And C, BZ, they are actually important for the chromosome mapping. Drosophila. There are so many retrotransposon linked to X chromosome as well as copia element, gypsy element, P element, I factor, they are also present. This P element causes hybrid dysgenesis, causes male sterility. Human, sign, the long interspersed sequences are there called line and short interspersed sequences are called sign. Sign has DNA and RNA polymerase 3 promoter sequence and line contains RNA pol 2 promoter sequence, apart from which ALU1 sequence and mite sequence is also present within the human genome. In other plants, we have Solanaceae foldback elements on 2 repeat sequence of the domain. Mechanism of transposition may be simple or conservative, may be replicative or retrotransposition. Simple or conservative. Here we can find they all have the transposase enzyme by which they make 2 cuts on the target site, having 3' hydroxyl end, which is the target for the DNA polymerase to extend them up and DNA ligase actually join them. So, that is actually the simple and conservative way. Next one, in case of replicative one, apart from transposase, another enzyme is present called as resolvase, because here the transposase make 4 cuts, and when the 4 cuts are made, donor DNA is co-integrate with the target site, and these co-integrate can only be resolved by non-homologous recombination with the help of resolvase, and in this way the replicative one is carried on. Next one, the retrotransposition. They contain integrase, reverse transcriptase and endonuclease. In case of LTR containing transposon or retroposon, LTR means long terminal repeats, where from DNA the RNA is getting produced, and from that RNA double-stranded cDNA is produced by reverse transcriptase, and both the double-stranded ends are sent by the integrase which integrate that particular cDNA to the host chromosome. And non-LTR containing transposon, what is happened? Here first one RNA is produced, acts as primer. Now endonuclease makes a nick after TTTA sequence in case of the host genome, and along with this the RNA is primed by AT primer. 80 base pairing. The classification are actually maintained by their mechanism to move. They may be class 1 retrotransposon or class 2 DNA transposon. So mechanism or mode of action is again actually how they are transposed. So first, the mode of action in class 1, they are activated or moved via copy paste mechanism. From DNA, RNA is produced, from RNA double stranded DNA is produced, which is actually getting inserted within the host chromosome. But there are some of the subtypes are there, copia, gypsy, etc. In case of long terminal repeat containing retrotransposon, U3, R and U5 are found. That is actually happen in case of copia and gypsy element. But in case of non-long terminal repeat containing retrotransposon, 5' untranslated region, then 2 ORF, next 3' UTR are present. They are actually found in case of sign, line and mite. Class 2, the mode of class 2 is actually propagated via transposase action. It is propagated via cut and paste mechanism. Okay, so at donor site you cannot find their presence, but in case of class 1 donor site the footprint is also present. Subtypes P element TN3, they are associated to the class 2 type of DNA transposon. Transposon and mutation. By faulty repair, destruction of the genome, dysfunctioning of the genome, they can create mutation. By exon shuffling and altered spliciosome, again the mutation can be introduced. Apart from which, RAG1 and RAG2 give the adaptive mutations and L1 give the inhibition of the transposition as well as transcription. We can find the telomerase is actually coming from the line sequences. So in this way, in all other aspects, we can find new combination of promoter, novel gene as well as new combination of the novel gene production by this sort of transposition mechanism. Transposon and mutagenesis, they can cause very clean mutation, maybe random or specific, and having 2 to 12 kilobase pair repeat sequences. After gene knockout, it is very easy to change the phenotype. Next one, transposon and C-value paradox. So transposon actually increase the DNA amount. So in case of maize human, we can find Arabidopsis thaliana, the increased C-value. Next, genetic significance. It causes gene inactivation, gene duplication, altered activation of the gene, recombination as well as differential gene expression. Now consequence, the novel gene is getting produced. We can have the restoration of the gene, null mutant allele as well as different other aspects of the transposons. Use in genetic analysis. They are used in gene therapy. The gene tagging is done. Sometimes the genetic analysis with respect to in situ hybridization is helped to locate the gene with the radioactive gypsy element. Next, medical significance with the RTF, that is resistance transfer factor by R plasmid. By conjugation, it can be associated with the host genome causing antibiotic resistance. So, TN4, TN10, they contain kanamycin, tetracycline, ampicillin resistance as well. Transposon and evolution. Transposon may come from the common last ancestor either or horizontal gene transfer. They can cause many aspects of the evolution related to the adaptiveness. They cause the VDJ recombination. They are regulated via integron attachment to the chromosome as well as they are caused for the sterility in case of P element, which is caused by the hybrid dysgenesis, as well as they protect the repetitive sequence, very novel, very useful gene sequence. And TI1 we know they actually are coming mariner sequence coming from the long evolutionary sleep. So that is very much important for the biological system. Transposition dynamics, it is related to the transposase action actually, and sometimes it is very low, and this transposase activity high frequency can be done by the environmental regulation. Next one, transposition is used as genetic tools because they are actually helpful to identify the transgenic as well as mutant allele. In case of Arabidopsis thaliana, so many genetic tools are actually used by this transposon. Sleeping Beauty is actually used to transfer or deliver genes in case of vertebrate system. Next, identification of the transposon. Because they are having repeats, all the repeats can be identified with the particular consensus sequences, and this is done by the algorithm which is KMAR. Biological significance, they can cause deletion, insertion, mutation and causes some of the diseases. What are they? The diseases related to hemophilia A, hemophilia B, the Doschner dystrophy, porphyria and several other diseases. Rate of transposition, their defense and induction. Usually TI1 mariner once in a month transposed, and we can find the disposition of the system in case of bacteria by deletion, and in case of eukaryotes by pi and siRNA the transposase action can be suppressed. But transposase have some promoter sequence which is heat shock, can resist very high temperature as well as heat and other environmental stresses. Advantages and disadvantages. Advantages means it acts as buffer against point mutation and acts as genetic tool as well, natural genetic tool for the protein and genes. And disadvantages, they cause chromosomal break, the break or disruption of the genetic activity. Now the virus are actually causing of the cancer. First, in 1908, Olaf found that the causative agent of cancer as virus. Then in 1911, Peter Ross found that Ross sarcoma virus is related to the cancer. Next, characteristics. So there are so many characteristics of the virus are there. They resemble to the prokaryotic transposon because mu phage, that is the longest phage, they have some transposase activity. Next, HIV-1, they have some property by which with the retrotransposition they can cause cancer as well. And we can find apart from these some lysogenic viruses are there. They integrate somewhere other within the chromosome, and when the lambda lysogens are excised out from that of the host chromosome, they causes excision or deletion of some of the sequences. Let's talk about their mode of action. So in case of mu phage, they are propagated via transposase action that I have described, making the cuts at the donor site and synthesis, causing the duplicated repeats. But in case of the HIV-1, they are propagated via retrotransposition, having integrase and reverse transcriptase activity. From DNA and the RNA is produced, from RNA double-stranded DNA to be integrated, they are produced. And we can find the lambda lysogens are propagated via the deletion from the E. coli chromosome, taking gal and biogenes. So in this way they resemble to prokaryotic virus and they can do the cancer. Biological effect. So by all these aspects they can cause the mutation, and such mutation and such deletion of the genes that is actually effective to cause cancer. Types: rubella virus, cytomegalovirus, hepatitis B virus, HIV-1, lambda, mu phages, they can all act as biological mutagens. Bacteria, they cause the oxidative damage. Characteristics: so by creating oxidative damage, they harm to the DNA damage to the host chromosome either, or sometimes bacterial plasmids can be integrated to cause antibiotic resistance to the host chromosome. Next one, the mode of actions. Mode of action may be propagated via DNA oxidation or DNA damage, and the DNA repair is suppressed as well as antibiotic resistance. When we are talking about the types, Helicobacter pylori causes the inflammation by accumulation of toxic agent within the stomach, and fertility as well as antibiotic resistance are found by the bacterial plasmid. Biological mutagen and environment. So biological mutagen causes disease, and severity of the disease can be changed according to the mutagenic power. So with some viruses it was tested. Analysis of the biological mutagen was done with inverse PCR and plasmid rescue technique. application, they are very much useful as the genetic tool. P element in case of Drosophila and Sleeping Beauty in case of vertebrate system help to transfer the genes, and along with the reporter gene, along with the marker promoter, they are actually transferred. In case of virus, it was found give the vertical gene transfer. In case of plasmid, they give the horizontal gene transfer. In case of symbiotic gene transfer, that is obligatory, but ordinary gene flow in case of higher level gene transfer. And transposon gives some sort of insertional mutagenesis. So students, I have discussed about many different types of biological mutagens, their different mode of action, their biological effectivity, as well as their relatedness to the evolution. Thank you.